It's a pleasure, although always a little sad because it means that her time here is up, to introduce Irina. I first encountered her, well, she says during open house before the term started during her first year, but I honestly don't remember her from that. But I do remember she walked into Biochem 8-9 the first day, which in that, remember, was a 2-term Chem 5 kind of thing. And Irina very quickly came to office hours and started asking me questions that had nothing to do with the material in lecture, but was really from the book. And I kept saying, that's really Chem 6. And then she'd ask another question. I'd be like, well, that's Chem 6. And that's Chem 75. and very quickly I realized that, she was operating at a very high functional level for a first-year student. I took her over to Professor Cantor's office. It might have been the very first time, and I stood there for about half an hour while they started going at it, and then I very quietly just snuck away. And I don't know how long they talked, but it was a long time. So that's kind of my first impression of Irina. When she expressed interest in working in my lab, I was sort of excited but shocked, and I said, you realize we do a lot of biochemistry and cell biology, and it's not very mathy. And she said, no, I want to do it anyway. And then she's really taken on a hard project in the lab, working with a difficult class of proteins. And she's approached her science, in a very deliberate and very, what do I want to say, skeptical way. So she's self-skeptical of her own results, and in a way skeptical of even the protocols that our lab uses in collaboration with Ron Taylor's lab, and has really sort of highlighted a number of things that, maybe we should do this better, or check this, or do this control. And, I've been very impressed with that. Her goal was to determine a structure of one of these proteins, and she's not quite there. I did everything I could do to try and entice her to stay for a 4+1 master's, but she said she's had enough.
I'm not sure that's how I phrased it, Professor.
She didn't say that. But she's going to move on and work at the NIH for the next 2 years before going to graduate school. So she's going to tell you about the progress of making and purifying this very ornery protein, and yeah, good.
I hope I'm on now, right? Okay. So as Professor Kuhl nicely introduced my topic, I will be talking today about VRF, which is a transcriptional regulator in a bacterium called Yersinia enterocolytica, and I will try to tell you a little bit about what I had done to characterize it structurally and functionally. So a little bit about the bacterium itself. It's a gram-negative bacterium. It's an enteric pathogen that causes the disease yersiniosis. And its virulence is regulated by the presence of the PYV plasmid shown on the right. The virulence factors can be divided into roughly 3 categories. Transcriptional regulators like VRF that induce transcription, factors that are responsible for the invasion of the host, and finally, secreted proteins that cause cytotoxicity and suppression of the host's immune system. So VRF being the major transcriptional regulator and sort of the trigger of the virulence pathway is, of course, the target for study and ultimately inhibition. So VRF is a member of the RSC family of transcriptional regulators, and a brother, so to speak, of another protein that our lab has been working on for a very long time now, TOX-T. So what you can see is a model done in the program Swiss model of predicted structure of VRF superimposed on the structure of TOX-T, which hopefully helps to highlight the distinctive architecture that is characteristic of the RSCs. The RSCs have a C-terminal DNA binding domain, and TOX-T shown here in blue and VRF in orange, that has 2 helix turn helix binding motifs, and then a spanning helix that runs and connects the two. This domain is largely conserved, as you can see from the relatively good overlap of the 2 structures. And then they also have a variable N-terminal domain that is responsible for dimerization and ligand binding. And that domain doesn't really overlap between the two proteins. So our hypothesis, I guess, very broadly speaking, was that being two transcriptional regulators of enteric bacteria that encounter similar stimuli in their invasion of the host, these two proteins might be regulated by similar small molecules. So we looked a little bit more closely into the ligand binding pocket that I showed to you on the previous slide in the N-terminal domain. And in TOX-T, which was crystallized with a palmitoleic acid bound in its pocket, I guess the carboxylic form of that, the head group is interacting through cell bridges with two lysines that are pointing into the pocket, but TOX-T has the lysines, VIR-F doesn't. The only charged residue that's pointing into the pocket in this putative pocket was histidine. So really the architecture, even though on the surface they might seem to be similar, was not found to be as conserved as we would have liked to see. The surface model of the two proteins with VIR-F shown on the left, the predicted model again, and TOX-T shown on the right, shows the two pockets and very roughly speaking, the differences between the two. So the TOX-T pocket looks much smaller and than the VIR-F pocket, suggesting that perhaps the ligands are different. So having established very, very roughly, using the simple analysis, that the structural homology exists between the two proteins, but it might not be conserved in terms of the ligand that might be inhibiting TOX-T and VIR-F. We wanted to move on and see if there was any functional homology associated with that structural homology. So as I mentioned previously, the two regulators encounter different stimuli in the environment as they invade the host, and one of them is bile. And different bile components, such as the fatty acids that you saw in the pocket of TOX-T. So the point was then to try to see if VIR-F, by any chance, could also be regulated by bile or a component of bile. So we used the beta-galactosidase assay to do that, and this is a very simple diagram chart that is supposed to help those who might not know what that is. So VIR-F, being a transcriptional regulator, has to bind to the promoter of the gene which it regulates. In this case, YOP-E was chosen. So this is the YOP-E promoter. This is a cartoon of the VIR-F dimer. And as it binds to the promoter, it induces the transcription of the gene called LAC-Z, which codes for beta-galactosidase, an enzyme that catalyzes the following reaction: conversion from ONPG to beta-galactose and orthonitrophenol. The production of orthonitrophenol can be monitored using spectrophotometrically. And if we take that to be proportional to the number of binding events, then we can quantify transcription. So this is exactly what we did, and these are the results showing beta-galactosidase assays with of VIR-F with 0.1% bile. They don't really show anything too promising. This is the negative control, this is the positive control, and this is VIR-F with, in the presence of 0.1% bile. And we can see that the inhibition exhibited by the protein, exhibited in the system is very mild, especially if we compare it to the inhibition observed for TOX-T. The next thing to check was the oleic acid, because that was found to be the specific inhibitor of TOX-T. So again, the result was rather negative in that VIR-F was not found to be regulated to the same extent by the presence of oleic acid, and we only saw about 30 to 40% full repression in the presence of oleic acid. Our conclusion was that oleic acid was not the specific ligand of VIR-F, but what we really wanted at this point, being a structural lab, was a structure. And so that we could make more intelligent assumptions about the ligand. So I guess I spent most of my time really trying to answer this question: how do you go from a piece of DNA to a crystal? And as Professor Kuhl has already spoiled the show, there are no crystals. I mean, these are not mine, pulled off the web. So I guess the outline would be first to clone the DNA into the vector of choice, express it in a cell line, isolate the protein from the cells, purify it, and crystallize it. And I will try to walk you through some of the steps that I did without going into too much gruesome detail, because it's not really the most fun thing about biochemistry, unfortunately. So briefly, I wanted to outline the vector that I use and some of its characteristics. VRF was expressed with a fusion protein comprising an intine self-splicing element and chitin bind domain. They are very similar in size. VRF is 31 kilodaltons, and the fusion protein is 27, 28 kilodaltons, which, on second thoughts, after having worked with a project for a month, or for half a year, I realized that that was not a very good thing to do, to have as a fusion something that can contaminate your protein and be of the same size and run the same size on a gel. But that realization came very late, unfortunately. So the chitin bind domain tag is used as an affinity tag for purification, and the intine is used to cleave off the tag here at the C terminus of VRF. Before I could purify the protein, however, I needed to express it. And I'm not going into details, but this little chart here shows all the expression attempts that I tried with IPTG. And all of them, I have to say, failed. The protein was either insoluble or it would just simply not express in large cultures. Finally, autoinduction was attempted, and it worked. You can see the fusion protein here shown in the pellet and in the supernatant, and then the cleaved protein, and the band corresponds to the chitin bind domain intine tag and to VRF. So all seemed to be going well until I decided to go a little bit deeper and see what the chitin column was really doing. And then I noticed several problems. First of all, this is a gel and a western showing cleaved chitin beads and chitin elution. And as you can clearly see, the cleavage reaction is not very efficient because a large amount of fusion remains even after 16 hours of cleavage time. Not enough protein is recovered from the chitin. And finally, even though very little protein is recovered, it's also contaminated with both the intine CBD tag and the fusion. So the column really fails to, it gets rid of all other proteins that you find in the bacteria, but it fails to separate the target protein here from its two contaminants. So I decided to fix cleavage first. Multiple things were attempted, and I'm not going into details because they are very boring, but I have to say that I looked into cleavage time, cleavage temperature, the identity of the cleavage reagent used in the reaction, salt concentration of the buffer, pH, and finally what really seemed to have worked was introducing a mutation into the C-terminal residue that is being attacked by the cysteine of the intine during the cleavage reaction, and that identity was key to the cleavage efficiency of the reaction. So after all of this, the cleavage efficiency was optimized, at least to an extent where it became productive to continue. The amount of fusion was reduced and the amount of cleaved protein was increased. The amount of chitin elution was also increased, but the contaminating CBD and fusion were still present in the elution, as you can clearly see. So my first idea of the solution was to simply pass the elution over the chitin column. Excuse me. I thought that that both the fusion and the chitin bind domain would simply bind to the column, and my VRF protein would flow through. And that did not happen because, unfortunately, I found out that cleaved VRF has affinity for chitin beads, and in fact stays on the chitin beads. As you can see, these are beads after the second pass of the chitin column, and they are enriched in VRF, as well as fusion and a tiny little bit of chitin bind domain, but nothing appeared in the flow-through, suggesting that VRF has as high of an affinity for chitin beads as fusion and chitin. Well, I had to go to a different chromatography step to separate the protein from its two contaminants. And so I picked sp column, cation exchange, because the PIs of the three isoelectric points, of the three proteins were so distinct that I thought that I would be able to separate them based on charge. In theory, I guess that would work, and in practice it didn't quite work the way that I imagined. So as you can see from this gel, this is the SP elution, and again, these are chitin beads taken to show the comparison between the two. So chitin beads contain the 3 species that I've been talking about, and the SP elution clearly shows that I managed to remove the chitin binding domain enantine tag, but the fusion was still very much present. So in order to remove the fusion, I try the gel filtration column, size exclusion, because the two, VRF at 31 kilodalton and the fusion at 58, I thought I would be able to separate them based on size. And so the initial elution profile looked very promising. It had this first small shoulder, which I thought would correspond to the bigger species eluting first, and then the main peak, which I hoped would be the protein, the target protein, VRF. However, when I ran a gel of the elution, it showed that the fusion was not restricted to this region corresponding to the main shoulder, but rather eluted together with the bottom band. I ran mass spectrometry analysis to confirm the identities of the proteins shown here, and in fact, this was the fusion and this was VRF mutant that I prepared. This was very surprising because I thought they were different in size, and the only conclusion I could make from this was that, in fact, VRF mutant was the same size as the fusion, and it was eluting with the fusion. The question then was, is it a dimer in solution? Running calibration of the S200 column, the size exclusion column, showed, in fact, confirmed the identities and molecular weights of the two peaks. And in fact, the main peak was found to be roughly 60 kilodaltons inside, which would correspond to a dimer of VRF, so about 62 kilodaltons. So VRF was found to be a dimer in solution, and as such, it could not be separated from the fusion contaminant within the limitations of our expression purification system, which was a little bit of a disappointment, because really what we wanted to do was to crystallize it. And to crystallize a protein, you really want it to be pure. At this point, however, we had done so much that it felt useful to just try and crystallize it and hope for the best and maybe assume that it would crystallize without being completely pure. So this is a table showing the crystallization attempts. Here is the concentration of protein in milligrams per milliliter. These are the concentrations that were tried, and these are the conditions that were tried. And the pluses do not indicate successful trials, they just indicate attempts. So none of the conditions yielded any promising results. At this point, I started having really, really bad thoughts because I couldn't quite figure out whether the protein was simply not crystallizing because it was contaminated, because of the presence of that fusion band that I showed you, or whether it was simply unfolded, inactive, and precipitating out in every single thing it could. So I decided to go back to the basics and check some things before proceeding to try to crystallize it again. So here shown is a CD spectrum of the protein, and it exhibits good fold with the characteristic peaks at 208 and 220 corresponding to alpha helical structure, confirming, in fact, that what we saw with the C-terminal domain with that had so many helices was actually true. And then the CD melting curves monitored at two temperatures, at two wavelengths, 215 and 222, show that the melting temperature was 41 degrees, so the protein was not exceptionally stable, but it should be stable at the temperature used for crystallization, so around 20 degrees. The next thing to check was whether the protein was active or not. As a transcriptional regulator, the protein was expected to bind DNA, and if it didn't, that would be a problem. So shown below here is the promoter region of the gene YOP-E that I showed you before for beta-galactosidase assays, on which footprinting assays mapped two binding sites, the strong binding sites shown in gray and the weak binding sites shown in white. So based purely on that, we expected that the protein would bind sequentially in a concentration-dependent manner to this piece of DNA that was cloned, first saturating the strong binding site and then saturating the weak binding site. The way that the binding was monitored was using ditch-labeled DNA, and that could be then visualized, and the shift of the DNA in the gel could be monitored. So this lane corresponds to free DNA without any protein added, and this is the maximum shift that exhibits on a gel. This is taken as a control. Now, I'm not going to go through all of them, but if we just focus on this little inset here, it shows addition of VRF protein in nanograms of protein per 20 microliters of reaction, and as we increase the concentration of the protein, we see the DNA shifted up as the protein binds to the first binding site, and the DNA migrates slower. Then as we increase the concentration, this shift becomes saturated, and finally the second shift appears over here that corresponds to binding of 2 VRF dimers to 2 binding sites of the DNA. So the protein seemed to be active, but there was another concern that remained. Was it really binding specifically in a specific manner or simply aggregating on the piece of DNA? And the answer came with another EMSA that was done. So again, you're looking at the DIG with the free labeled DNA here as control. This is simply a titration of protein into DNA. And then what I would like to focus on are these two insets first. So the premise of a chase-off assay is to take a piece of unlabeled DNA added to the reaction in increasing concentration and see if, in fact, it will outcompete the labeled DNA and unshift it back. So here you can see that protein shifts the DNA up. We see the 2 shifts corresponding to binding to the 2 sites. And here, in the presence of increasing amounts of unlabeled specific DNA, the shift is recovered, which indicates specific binding. Finally, as a final control, we cloned a non-specific piece of DNA that was not supposed to bind the protein. And in fact, in the presence of that non-specific DNA, regardless of the concentration, the shift still appeared. And there was only very mild recovery of the initial shift. All of this was to point out that VRF, even though it did not crystallize, was active, folded, and it did what it was expected to do. Finally, because I had lanes left on this gel, I tried to see if I could inhibit VRF with the molecule that was specifically designed for TOX-T, an inhibitor of TOX-T called ristatin. And it seemed to me first that maybe at 5 millimolar, ristatin was really shifting all the DNA back, and so binding was inhibited. But then when I looked at the DMSO control, which is the solvent that was used for ristatin, it looked that it was simply that the solvent might be just disrupting binding. So really no significant results about inhibition yet, but I think the important point was that binding was observed and binding was specific. Finally, I wanted to briefly touch upon what these findings suggest about VRF and the DNA binding model that was devised for TOX-T. So when the TOX-T paper was published, it proposed the following scheme in which the binding of a fatty acid that I showed you previously in the pocket, the palmitoleic acid or oleic acid, would disrupt the formation of the dimer that is necessary to activate transcription. And it was hypothesized that the dimerization was happening on DNA. However, the fact that VRF was a dimer in solution and bound DNA as a dimer rather than dimerized on DNA might suggest that this model, at least for VRF, might not be quite correct. And we would want to spend more time thinking about the mechanism of VRF binding to DNA and regulation of transcription. And I think that at this point, I'm just making my final quick conclusions. So what I tried to do, at least, was to test several compounds using beta-galactosidase assays, and the results were negative in that they didn't show regulation similar to the one observed for TOX-T. Then VRF mutant was successfully expressed, purified, at least to the extent that it could be used for functional studies, and shown to behave as a dimer in solution. And finally, it was characterized using CD and EMSAs and found to be active transcriptionally. future directions, of course, involve crystallizing the protein, because this was the goal from the very start, and this was the goal that was never achieved. And then, of course, continuing to characterize the transcriptional activity of VRF. And I guess I will just finish by thanking Professor Kuhl for having so much faith in me that he gave me complete freedom to go on a wild goose chase hunt of what I was trying to do, and just making this very much my own project. My wonderful lab members, and Brittany unfortunately is not here, but I would like to thank her for all the support and all the help that she has given me in those years, for reading my thesis so many times, for becoming a friend, Annie for everything, including the wonderful EMSAs that you saw that worked the first time I tried them and worked every single time. And when you find a scientist who gives you a protocol that works the first time and every single time, well, latch onto them because they must be really good. And then I would like to thank Charles, who's a postdoc in our lab and who has taught me, together with other graduate students, everything that I know. I would like to thank Gabriella, who helped me, taught me, worked with me on every single microbiology experiment that you saw on these slides, and on many more that I didn't show that failed, or will not go into that. I want to thank Michael Stevenson for helping me with DSC, for helping with CD, for answering my questions and being very supportive of my efforts. I can't think, I have so many graduate students to thank that I ask you to forgive me if I don't do that publicly, but I will definitely do that personally. I want to thank Anda and Karen, who are not in my lab, but who Karen helped me with gel filtration, and she explained to me in 5 minutes how to do that, the procedure that I was doing wrong for a very long time before I ran into her. And Anda helped me purge the FPLC when I drew air into it and was in complete panic. And I can't even name all the things that they have done for me. I would like to thank my thesis committee. I would like to thank the chemistry department for taking the time to make this really a home for me, and really tolerating me for the past 4 years. I don't really know. I'm sorry, I'm getting really sentimental here. So I'll just finish off by saying thank you to the undergraduate Office of Research for supporting all the hopefully not crappy research that you saw right now. Thank you.
Very nice. Any questions? Gordon?
Yeah, did you try Annie's inhibitor of toxicity against your protein?
Yes, so no. So I just, no, when you saw that EMSA with a porphyrostatin pressed kind of to the side, these were, these were the last lanes that I had, and that was the last EMSA that I ran before I started my thesis. And I think it would be very appropriate if Annie, if she really wants to take on this protein, test her compounds on it, because it is very much her project.
Mike?
So your size exclusion.
Yes. Would you like me to flip back to it?
No.
You have it stored in your memory? Yes.
So often working with dimeric complexes where you have a mixture of the uncleaved and the cleaved, you can end up with these heterodimeric complexes where half of the protein is cleaved and half is uncleaved.
Yes.
Do you know if that's happening in this situation?
So what you're referring to is you're saying that that VRF cleaved could be also complexing with the fusion, because the N-terminal domain, I guess, is freed up. I mean, in my thesis, there's a table that lists all the possibilities, dimerization of the two fusions, dimerization of the two proteins, complexing between a fusion and a dimer. And I don't really know what a very good way to quantify that would be, because the gel, you boil it in SDS and you run it in DTT, so you completely, you do away with any possible bonds that could exist between those complexes, so what you end up with is just the relative amounts of the two, and I suppose you could run a native gel, but I really didn't do that. And so I guess my way of trying to deal with the situation was to simply remove the fusion, and then you would know that you're not really complexing with anything but itself, at least. I don't know.
I have 2 questions. I'll ask them one at a time. So,
please do.
my first question is, in your purification strategies, have you tried a precipitation-based method, like with ammonium sulfate?
I did not. Does that involve refolding?
It wouldn't denature it. Presumably, it would stay folded.
I have not, unfortunately, done that, and maybe I should have. I think that I was trying to do my best within the system, which when it turned out not to be fully functional, I did not have the time to switch to a different one, but no.
Okay, and my second question is, have you considered doing maybe an affinity column with a DNA piece that it perhaps bind to kind of pull it down selectively?
I did not think of doing that. I was hoping to maybe simply complex the protein with DNA, pull it over put it over gel filtration, isolate the complex, and then try to crystallize that instead of the protein by itself, hoping that the fusion would not bind. But the problem is that the fusion still contains the VIR-F part, so it would, it might also bind, even though it's not a dimer, it might still bind DNA. No.
Final question?
I was wondering if it's practical to inhibit dimerization during your gel filtration by adding a fatty acid to your overfitting.
It would have been very nice if if we had a fatty acid that would bind the protein and inhibit dimerization like it does in toxicity. I have not done enough beta-galactosidase assays with other components of bile to know if there is a fatty acid that does that. And I was actually trying to, so it would have been a good one if we had a good inhibitor, which we still kind of don't. The other thing that I was thinking along the same lines, trying to prevent dimer formation, I tried gel filtration at different salt concentrations, because I thought it might be, just like a salting out effect or something, and not a real dimer formation. But it seems to be a very strong dimer. I'm also very, very afraid of adding anything because it's so insoluble as it is and unstable that if I just start, pouring in DMSO and hoping that the dimers fall apart, then they will probably fall apart, but then the protein would too.
Okay, please join me in thanking the